Hello, everyone. My name is Charlie. I'm one of the co-founders and CTO at Parse, and I want to thank all you guys for coming out here to learn more about automation. We're going to go ahead and kick off the kind of presentation section of the day. And so you'll be hearing from myself, Devin, Hoy, and Ashling, talking about various different topics that Ashling kind of talked about in the beginning. And so I'll kind of go ahead and start off with kind of quick overview of the company and technology and just talk about single cell generally. Actually, first off, uh, okay, great, there you go. Awesome, cool. Well, first thing that's probably worth talking about, just taking a step back from like, um, Evercode technology or Parse generally, just looking at single cell, the field and how it's been evolving over time. Um, if you look at basically publications that have been coming out using single cell sequencing, basically it's just an exponential increase of how many cells are being profiled per study or in this case, per publication. And it's not so surprising, actually, if you look at that same kind of data set here of all the different publications, you'll see that looking at how many cells are being profiled in a publication is pretty correlated to how many cell types can be resolved within that publication. And so it kind of what that means to me and what it's kind of telling me is that we probably don't understand biology in quite as much as we might think we do, and that usually it's more complicated because if you just profile more cells, more biology is being resolved. So it's not like we're hitting a ceiling or anything of just saying we've done enough cells, now we're good. And I think it's kind of being underscored by some other things that are happening in the space right now. So there's this preprint that came out a few months ago from authors from Sanofi and the Sanger Institute, where they looked at basically different drug regimens. They looked at basically 30 different indications across, I believe, 13 different tissue types, and just try to look at, they kind of asked the question in a retrospective way, if we had gotten single cell sequencing data or single cell RNA sequencing data in particular here, what would have happened over the course of these studies? And their conclusion across this study here was that if they had leveraged single cell RNA sequencing data, they would have actually tripled the chances of getting a target through the phase 3 trials, which is a significant difference, obviously from time costs, people costs, and money costs as well. And I think another thing that we've kind of seen happening in the just generally around the world right now is this big evolution of AI and how we've all seen ChatGPT coming up and becoming more popular here. And it's kind of worth pointing out, like, how did all this happen and how could biology possibly intersect with all these other things happening in more of the tech space? And really, the precipice of what happened with, things like GPT coming out was that they had these AI models called transformers. And effectively, they didn't really work out until you had enough data. And there's this like inflection point where once you had enough training data to put into this, all of a sudden things were working really well. And that's basically underscored by this plot here, where the y-axis is just looking at number of training data points. And so the question is, like, can biology ever get to that point? And we'd obviously like to argue we can. And I think the way you can do this is by leveraging single cell RNA sequencing data, because you can get lots of different data points at the single cell side, but then also leverage the parameters per cell, say 20,000 or so genes. And you can really start learning things that we didn't even know we could learn by getting enough of that kind of data and putting it into these models. And so that's basically exactly what Parse is trying to do. Where all these trends that are happening around the world right now, we're really trying to basically make single cell sequencing much more scalable and much more easier to do to actually help people just do these larger scale studies that we can see are kind of needed and in high demand right now. And we're really humbly working now since launching in 2021 with over 2,000 different labs around the world. And really, more importantly to us, it's not just that people are using the products and the kits that we were selling here, but actually that people are generating really high quality data and a lot of it too. And so here's just a few different kind of showcases of different customer data sets that we have hosted online. That it's kind of a small subset of the 100 plus different publications and preprints that have come out using Evercode technology. At a high level, kind of what's what are the main advantages and why did all those people start using Evercode? There's kind of four main things. And so first is really scalable. Right now you can go from basically 10,000 to up to 1 million cells or nuclei within a single run. And that can be leveraged up to 96 different biological samples. We've done a lot of work to improve data quality. So that's mostly talking about kind of sensitivity, number of genes or transcripts you can detect per cell, as well as kind of not having impurities like other technologies have, like ambient RNA. As some of you may know, we don't require any custom instrumentation, but what that actually means is that we can become really compatible with a lot of the instruments that we've been looking at today. And so there's no kind of black box where we can't be continuously innovating and releasing new protocols as we also innovate and release new kits. Finally, everything we do is actually leveraging fixation. So we actually fix samples up front. That's basically the first step of every one of our workflows. And what that means is you can actually fix samples in one location at one time point, or really whatever kind of setting. It could be at 5 p.m. on a Friday. You don't really want to keep doing more work at that point. You can basically lock in your biology at that point and then run the assays later on. And so coupling that with a really high multiplexing ability of say up to 96 samples means that you can store samples over time or over different locations and pull them together into single batches. So this is kind of the high level workflow or solution here. Starts out with fixation, as I mentioned, barcode library preparation, which is a lot of that's we've been talking about automating today. Then you would go into sequencing, where we are compatible with most sequencing solutions that are out in the market today. And then finally, we'll go into data analysis, where we have a really great solution called Trailmaker that some of you may have already seen, and Ashley will be talking about more in just a few minutes. So at a high level, the way the technology works is we actually start with the same single cell suspension you would start with for any other single cell technology. And rather than introducing individual compartments like oil droplets or microwells, where you isolate one cell in one compartment, break it open, and then do biochemical steps within that one compartment, what we do is we actually use a cell itself as a container for its own barcoding. And we do that through this fixation and permeabilization step. So that's basically what we're showing in this slide here. And essentially what that means and what that means we can do is we're actually kind of locking in the RNA or other molecules inside the cell. But then we're basically chemically poking holes throughout the cell wall to allow barcodes and enzymes to kind of flow across the cell wall and act on, say, the RNA. So with that kind of step that's occurred up front, we can then subject cells to what we call split pool combinatorial barcoding, where we take cells and that have been fixed and permeabilized. We split them up into a first 96 well plate, where every well in this plate has a kind of unique well-specific barcode. And so if you say zoom into one of the wells here, you'll see this blue well here. There are many cells in that one well, and there's basically an in-cell reverse transcription step occurring. That's basically at the same time creating cDNA and also appending this first kind of well barcode. And so we're not uniquely barcoding any cells quite yet here, but what you can maybe see is if we actually were to say pull the cells together and split them up into a second plate, where the second plate also has well barcodes associated to them, you can maybe begin to appreciate the cells are now starting to take a unique path in this split pool process. Or if you say zoom into the yellow well here on the second round, this cell on the top left, say, came from a blue well in the first round, whereas a cell on the kind of the right came from a pink well in the first round. And so very importantly, this unique path that's kind of occurring through this process is actually being documented via the combination of barcodes at the end of the transcripts. And unlike, say, droplet-based methods or microwell-based methods that scale linearly as you add more compartments, split pool barcoding actually scales exponentially as you add more rounds. And so here, this is kind of the true configuration of our mega kit, our 1 million cell kit, where you can actually generate on the order of 14 million total barcodes, or 14 million possible paths a cell can follow in this process. And so what that means is if you're actually barcoding, say, 1 million cells, the chance that any 2 of those 1 million cells follows the exact same path is actually quite low. And so a way to measure that, basically we're showing you this way of doing single cell sequencing that doesn't ever isolate one cell in one compartment. And so just to kind of prove that to you all that this actually does in fact work is doing these really standard kind of quality control experiments in single cell sequencing, where you basically mix a mouse cell line and a human cell line together right at the beginning. We fix, then run the entire workflow, and essentially give every perceived cell the opportunity to align the reads to a mouse or a human genome. And so effectively every dot here on an axis is one perceived cell. And you ideally want to see every dot on the axis because they tell you have many reads from one species and none from the other. And so anything in the middle here would be basically a mixed or a doublet. So we see is actually the doublet right here for our 100K kit is quite low at 1.7% observed. And we look at basically other size kits, our 10,000 or 1 million cell kit, we basically see very consistently low rates, really across the board and much lower than basically other technologies that are out there. Do you want to really emphasize the idea of fixation? I talked about this a little bit already, but basically being able to kind of fix samples on different days and different times has been super, super helpful in us logistically making single cell much easier for our customers. One other tidbit I'll mention is that you can not only fix samples and ship them around, but you can also fix and aliquot the same sample. So if you say want to run one sample immediately here, but you might want to do like a smaller run first, and later on you realize you actually want to get more cells from that same sample, you don't have to go back and redo the whole experiment. You can just take another aliquot of that same fixed sample and just run it again. And of course, we've done a lot of studies here just to make sure data quality isn't impacted here. So kind of the key axes there for sensitivity is transcripts per cell and genes detected per cell, where we get a really similar kind of sensitivity even after 6 months of storing those fixed samples. Yeah, we have released our newest version 3 chemistry here a few months ago. And so what that's been essentially done is shortened the workflow pretty substantially here, while also at the same time increasing gene detection. And so you kind of see our evolution from the different versions here. We're just basically continuously going to be improving this sensitivity for the entirety of our company lifetime. And with this assay, we've also basically reduced the total amount of time it takes to actually run it, which obviously also means automated workflows will run faster as well. Just to give you one more sense here, just kind of tidbit of like why scale matters. Here's just a really simple experiment. We did 1 million cells across 12 different PBMC donors. And kind of looking at what the heterogeneity was here. Here we can just look across 12 different samples, see distribution of different cell types that we were able to collect. This is, again, we got all in a single experiment. And one thing that's kind of worth pointing out is if you start looking at kind of some of the more rare cell types here, in this experiment, doing 1 million cells over 12 samples, you're going to get about like 80,000 cells or so per donor. And with that, you're actually able to start to detect really rare cell types. And so, here you can see on the axis, out of those 80,000 cells per donor, we're getting 100 or just tens of cells from those individual donors that are being detected. And some of these are actually quite important, like innate lymphocyte cells or stem cell progenitor cells. If, like, for example, you were to do a more traditional single cell run, like a few years ago, you say the scale is like 10,000 cells per donor, that would basically be under the limit of detection, and you wouldn't even be able to reliably call some of these cells here. And so I think it's a really, really simple example. It just underscores a need for scaling up single cell. So with that, just want to give you guys a high-level overview of basically everything we currently offer. Basically, 3 different size kits, as I mentioned earlier, between 10,000, 100,000, and 1 million cells. And this actually can be done across different modalities here, getting just gene expression. We also now have immune profiling, so looking at either T cell receptors or B cell receptors coupled with gene expression. We've enabled single cell pooled CRISPR screens, and also gene panels, which allows you to kind of select for only a subset of genes after you've done kind of the whole transcriptome assay. And so, yeah, if you guys have any questions about the kind of high-level technology here, feel free to reach out to me or anyone else wearing Parse shirts after the talk, and we're happy to talk more about the technology and the chemistry as well. With that, I'm going to hand it over to Devin to talk more about the benefits of automating Evercode in more detail.
How's it going, everyone? Devin Davenport. I am a biochemist by degree, but a automation engineer at heart, and currently a senior automation engineer here at Parse. I'm going to be talking about the benefits of automating Evercode. Um, so as Charlie mentioned, it is a combinatorial barcoding uh, proprietary system that we use here. And Parse is in a good phase right now where we are initiating the automation of our workflow to help facilitate researchers and labs run the Parse assay in a streamlined manner. And it's an exciting time to be at Parse because we are automating. Why automate? One of the main reasons I got into automation, and most people like automation, is because it's predictable. And when you can predict what's going to happen, you're more likely to invest in those experiments because you have no fear of losing sample, money, or time. So automation essentially can streamline manual repetitive tasks to produce high quality results and minimize that variability in experiments. So this particular assay is a 2-day process, right? And so you have an analyst that performs the assay day one. And then day two, they're not feeling so well. You know, the variability in their normal throughput has gone down for whatever reason. It's variable. You can also have an analyst take PTO, and then day two, another analyst picks up and finishes that assay. There could be some variability there. But if you have automated it, it doesn't really matter who executes the button pressing on that instrument. You're going to get that same data output, or at least you should, right? And so that's what we like about automation. First and foremost, is that you can't predict it. You're going to be able to consistently achieve the results corresponding to the data generated by your non-automated workflow. So that means essentially, once you hone in on the automation, it is consistently going to output what you expect without any question of variability. And if something were to happen, automation allows you to essentially trace back, see what happened, where did it go wrong, and you can kind of analyze how to troubleshoot that. Whereas a human performing that task, you say, well, what happened? The human analyst is going to say, I don't really know, or they forgot. And usually as an analyst, when I was doing the bench work, I'd be doing 384 well plates, get to a part and say, did I add that? And that question right there is something that automation takes away from the analysts having that burden of focus, right? And that's what we want. Um, so essentially, the big part about this is it's scalable. Okay, so you can do the same thing multiple times and keep growing simply by purchasing another instrument. Transfer that tech on that instrument. The method's going to perform the same way because it's the same instrument. So you can take a series of molecular biological laboratory steps and reduce it to the press of a button. And that is powerful because it's easier to scale with predictable throughput. You can operate efficiently and accurately for extended periods of time. Like I said, when I was doing 384 well plates, I'm at plate 13 and I lost focus. Where am I at? Is it ruined? That's a bad problem to be in, trust me. You can re-engineer the process. So with automation, as you see all the instruments here today, as they come out with new versions, the tech is getting better. So all you have to do is upgrade, right? And now you're getting better performance overnight. Whereas it's very hard to increase the performance and throughput of an individual. You essentially have that individual skill set and you're relying on them to perform at a certain degree over time. A robot can be upgraded at any time, software and hardware. And then as time goes on, automated workflows become easier and performances continues to be optimized. So again, the trajectory of automation is going up as technology advances, AI is getting involved, everything is becoming easier to automate, and we want to help facilitate your ability to run Evercode in your lab conveniently. So as we've automated the Parse Evercode workflow, we've essentially been able to reduce the hands-on time by over 75%, especially the pipetting steps, over 85% reduction. We've totally moved all the liquid handling onto a platform. with Integra, Opentrons, Hamilton, SPT LabTech, all of the pipetting steps are essentially done by a robot. So you have that accuracy, you have that confidence that you are transferring the amounts, the volumes that you need, and getting the reactions as precise and accurate as possible. The last thing is cost effective, right? Automation, as you scale, you're going to need less personnel, right? Because the instrument can essentially do more samples, or you get multiple instruments and it's going to outproduce humans needing to be there, or you need to train that analyst to be able to run that biological workflow. Whereas with automation, you get an instrument, you train someone how to push the buttons, and then anyone can come in and step in for you. The analysts, again, they don't need to be trained in the molecular biology techniques. You just simply need to be able to follow instruction, visual instruction. A lot of the GUIs that you've seen today make it easy for anyone to go over there, press a button, and kind of get started. You can conveniently mitigate human error. I think that's what we're all trying to avoid, is human error. And again, as you're running a method of any type, all it takes is one loss of focus, and now you're questioning the entire experiment, right? Whereas automation kind of alleviates that burden, that pressure of knowing every step and where you're at, and making sure you don't make any mistakes. This is really just a convenient way to mitigate, again, that human error. So you can significantly increase productivity and overall throughput with automation, and that's really the main goal here that we want to relay to you all, is that we're here to help you facilitate getting Parse Evercode working in your lab and running effectively and efficiently. So the justification, again, is predictable, scalable, and cost-effective. You're going to have minimum variability with increased throughput and reliable results. You'll have a predictable turnaround time. When is this going to be done? You know exactly when it's going to be done, because it always takes 3 hours and 40 minutes or whatever. You know when that turnaround time is, and that's very powerful for labs, researchers, and all the likes. You'll have low failure rates and less repeats. Again, the traceability is a main thing for a lot of labs, knowing what happened when. You're generating more data, and you'll have an optimizable process. So over time, it's going to get better as more things are innovated. You're able to quickly address those and upgrade your process. As you see, we have partnered with automation vendors, currently Integra, Opentrons, SPT LabTech, and Hamilton. There will be more in the works, but again, we are just getting started. We're excited to get these automated solutions out for you to use and try. And the thing is, it's a variable system front that you see here. Depends on what your lab needs. Do you need tight space, less real estate? We've got an Integra, very, very tight spaces that can do everything you need. If you have the space and you want to scale, Opentrons, SPT, Hamilton, these are, modularly getting bigger for your lab needs, and you can scale with those as you need. So we have many solutions for you. And again, we're going to keep partnering with other vendors to help make sure we can address any and all your needs as you want to automate the Evercode process. So to that note, I'll be passing it off to Hoy, who's going to discuss and tell you about the tips and tricks that we've encountered as we've automated. And yeah.
Right, thank you, Gavin. All right, hi, I'm Hoy. I'm the Senior Automation Director here at Parse. With my stint, I've done a lot of automation workflow with NGS, primarily at Color. I was doing somatics cancers, and then at Resolution, I was doing liquid biopsy. And here I am at Parse now doing single cell. So just as a background, when I first decided to join Parse, I looked at our protocol. I'm like, yeah, it's easy. I can probably do it in a month. And lo and behold, that was not the case. Yeah. So hopefully with this tips and tricks, we can get you moving a lot faster and transfer the knowledge that I've learned for the past 2 years since I've been here at Parse. So just, I think the one powerful thing about Parse's chemistry is that we are instrument agnostic, meaning like we're able to work with any liquid handler, any automation to get this process working efficiently. And our aim is just to help researchers and labs scale up their experiment in order to generate more reliable data through automation to advance the understanding of single cell biology. Whether it's your lab is doing like 10,000 cells or millions of cells, we're here to help. And that's the key thing. So just as an overview, I'm just going to cover basically the tips for setting up like successful automation and then dive into what success looked like. So the first thing that I've learned here at Parse is like coming from a background where I've never worked with cells before, is that cells like to be cold. You know, and I think my first stint here trying to automate this was like, yeah, let's just do it at room temperature as fast as possible. That didn't go very well. You know, because ultimately there's RNA in these cells, and RNA is not stable and degrades very easily at higher temperature when they're not frozen. So what are some of the things that you can do? The first thing that we did was either, you know, we added on active or passive cooling positions for our automation. And also like we need to like pre-chill a lot of like the equipment and consumables in order to make sure that the cells are happy where they want to be. And here are some of those solutions. For active and passive cooling. As you can see, the first image here is what we've actually custom designed, what we call our ice block strategy, where you can either fill this with ice and store in the -20. And then right before you run start running your process, you take it out. And it's very critical because since now it's like, you've just taken out the -20. If you put your plate on there immediately, your cells are going to freeze and bad things happen. So just as like for ours, we recommend like leaving this outside at room temp for 10 minutes prior to use. So just so that it can warm up a little bit. Ideally, your cell should be like 1 to 4 degrees. And then these are the various ways. The second way strategy is using like liquid water circulators. The benefit to that is that you can like, it can cool multiple positions, lots of positions, right? But the downside is that you do need to wait a little bit longer in order before starting, because it does take roughly half an hour for your systems to pre-chill. The last one is the Peltier coolers, such as the Hicos and the Oventron cooling modules. Those take a little bit less time, 15 minutes. But once again, you know, like it's very important to let these get to the proper temperature before starting. And if possible, anytime your containers like touching the cells, make sure they're kind of pre-chilled before starting also, because if you just take it out and shove it on to, you know, whatever device these are, they're not at the proper temperature. So you're going to have that large temperature fluctuation, which can reduce your performance. Tip number 2 is that minimizing the amount of time that cells have been after it's been thawed. And it's very critical. This is like a hard set lesson for me. RNA is prone to degradation. That's what I say when I work with single cell. And so we recommend like making like smaller sub-aliquots of your fixed cells just for counting. Because even though there are automated cell counters, if you're running like 96 samples, that can take like anywhere from half an hour to an hour. And if you're counting by hand or manually, or by, you know, like using a person, I know for me, it takes me like at least 15 minutes to just count one sample, right? Because you're counting 4 quadrants, and that's not very fun. And then the other thing that we recommend is like possibly like using the smaller sub-aliquot, counting your cells, and then preparing your normalization plate beforehand, before thawing your cells. So that way you can just, once your cells are thawed, you immediately transfer that and then get going to the barcoding round. And like with automation, depending on your automation platform, you definitely want to optimize the pipetting patterns, right? Because most of the time, liquid handlers like to do things in columns. And so you want to do that along with possibly splitting your samples, right? If you have 8 independent channels and you only have 1 tube of sample and 1 channel can only go in the tube at a time, that's going to take 8 times longer versus if you're splitting your samples, it can pipette all 8 positions at a time. And then more importantly is that unless there's a safe stopping point, definitely proceed to the next step. So here's tip number 3, cell straining. Surprisingly, this is like, you're not the only one out there that has this problem. There's actually a lot of clients that run into the same issues, and we ran into automating this as well. And I don't have a PhD in single cell, and no one told me this, but apparently there's different sizes for strainers, and size come in all shapes and sizes. So definitely, if your cells are getting clogged, maybe the strainer is not the appropriate pore size. And then also dispensing cells to a strainer is actually pretty counterintuitive, at least for me. You would think that you want to pipette in the middle as fast as possible. But that's actually not the case. At least that's what we've learned sometimes. Sometimes you need to dispense to the side of the strainer and then dispense very slowly. What I believe it is my theory, is that slowly dispensing it, there's a wicking action on the side that helps drag the leftover cells down into the tube. And here's a video of, I guess, the first time that we've tried to do it. And I just want to point out, let me play that again and pause it at the right moment. This is where we've kind of dispensed as fast as we could right in the center. And what you can see is that there's lots of bubbles that's forming on top. And I think a lot of clients are running into this issue. So, so this is this is how we solve for it, by offsetting the dispense speed, offsetting the dispense positions, and reducing the dispense speed. We didn't use all the tips in this case, but we just used 4 for testing. But as you can see, there's actually cells in these samples. We kind of added some food coloring, but now you can see it's dispensing very well, and then there's no clumping on top or bubbles on top. Number 4, mix vigorously during sections 1. This is kind of counterintuitive for me too, as an automation engineer, in that I always thought that cells are fragile, right? If you mix it really hard and fast, they're going to lyse and you have no more cells left. Luckily, with our fixation protocol, I think it stabilizes the cell membrane a little. And after watching actually the R&D scientists perform this manually, I'm like, whoa, you are pipetting really fast. And so that's what we started doing, and we've gotten so much better data. Where I think it's improving is that your cells are probably adhering somewhat to the plastic. So if you're aspirating, mixing softly, gently, you're not getting those cells off those plastics. Number 2 is that larger cells, if you're not mixing fast enough, they tend to settle. So your first, if you're not mixing adequately, your first transfer will probably have no cells at all. Whereas your last transfer, it's going to have all your cells. So that's what we've seen also. And then the third thing is that for automation, at least, my number 1 pain point in general for automation is mixing. I have not found a better way. And usually with chemistries, you want everything to mix very thoroughly. And if there's any problems in your assay, normally it pinpoints to mixing. So, and then also as an automation engineer, I didn't know this. So the first time I tried to automate this, I was like, hey, let's vortex the cells. And no one told me this. I was like, hey, I can do this faster, and that wasn't the case. So do not vortex your cells for barcoding. Bad things happen too, because cells start slicing. So tip number 5, these are just miscellaneous stuff for preserving, increasing your cell retentions for sections 1. There are 3 centrifuge steps is it 3? Yeah, three centrifuge steps for our protocol. And normally after the centrifuge, the cells are pelleted. After it's done, you want to move quickly as possible from the centrifuge to your next process. The point being is that cells tend to float and come into solution, so you're losing cells that way. And also it's kind of a good balance too, and it's one of those challenging things that people don't think about. Okay, so you have your cells floating away from the pellet, but you don't want to accidentally pipette the pellet. So what would you do? So how we tackled it was the first initial couple large volume removal will actually go pretty fast. But as the surface, as the volume is closer to the pellet, we'll slow down our aspiration speed so that we don't disturb the cells. The next learning thing about that is try to reuse the tips. As you know, every time you make a transfer, there's probably like some samples that stick on the side of the tip or actually inside the tips that get tossed. So you're losing cells that way. We also recommend using plastic that is only polypropylene, especially when it's coming in contact with your cells. And if possible, if your pipetter has low retention tips available, definitely use that. Bead cleanup is actually really critical for our assay, actually for any NGS workflow. And it's actually really critical for our assay too, because there's a lot of washing steps. And these are the recommendations for the magnet modules that we've used. The first one being the alpaca. The second one is basically made by Opentron, and the last one is Integra. I think there's two ways. The first two are like this ring pattern magnets, whereas the Integra is it's a corner magnet, so your cells will only attach to one corner. And each one of those has its own different problems, right? So tip number 6 is minimize bead loss, especially for sections 2. Because if you think about it, all your unique molecules are bounded to your bead. So any bead that you're picking up, aspirating, discarding, you're losing those unique transcripts, which tend to lead to lower library complexity. So here's a question for you guys. So what's wrong with this image? Okay. Okay. So okay. So this is placed on one of the corner magnets. As you can see, the super name is not very clear. So it's slight tint of brown still. So if you were to remove this right now, that means you are discarding beads. So, like the strength of magnet and how the volume of your sample plus the proximity of the magnet will impact how long you have to wait before the super name removal. One way that we've gotten around this, if it's too much volume, you can actually do a simple aspiration and dispense to get the samples closer to the magnet, improving recovery. Also, one downside to this type of magnet is that your cells are clumped together more strongly at one position, so resuspension might be an issue, right? So for this, you might have to mix a lot more. The second option is the ring magnet. That has another problem too, is that these ring magnets, can you identify what's wrong with this? I know it's a little small, but there is a little bead speckle at the bottom of the plate versus where the ring is supposed to be. How we've gotten past this is we kind of do like we use the eye swap to basically wiggle the plate a little bit, so that helps get the beads a little bit higher, so that you don't accidentally aspirate the beads. Or the best solution that we found so far is don't do pipette mixing, right? Vortex. Because these are no longer cells. These are just cDNA. So they're stable, just vortex. And that's what we actually recommend for sections 2. What we recommend is basically heating and shaking modules. And that's what we've included. We try our best to include it. The heating modules should be able to go up to 37, and most of them can go up to like 80 or 100. But for our process, we only need 37. And shaking modules should be able to shake like up to 1800 RPM. And actually, like, if you are just constantly mixing using the heater shaker, it doesn't actually work very well. So we've added this pulsing step. So for the shaking, it stops, it starts again. It actually switches directions just to aggregate the beads and mix it a little more thoroughly. And then tip 7, general best practices. This is just lab stuff. But basically, before you start, definitely wipe down your benches and equipment with RNase prior to starting fixation in sections 1. Robots don't like bubbles. and droplets on the side of the tube because it can't get to it. So we definitely recommend like centrifuging these the reagent tubes. Some of the reagent volumes are pretty small, so centrifuging definitely helps get the reagents and enzymes where they're supposed to be. And then third, place seals or lids on top of your open containers, especially for these long incubation steps to reduce contamination. So I'm going to go over what success looks like. The powerful thing about Parse is that our fixation like protocol, right? Like we can fix cells, store it, and use it later. And that's the important thing. Once you get data on the first fixed cells, you can actually use it as controls to monitor your automations platform over time, especially for labs that are, CLIA and FDA that are following like CLIA and FDA guidelines. You need to do those competencies and validation studies. And with our fix, we can do a lot of cells at the same time, up to 96 samples, especially with the plate-based fixation. And the third thing is that we actually have our own controls that we can help send out for your development, that we know the sequences, the sequencing metrics for those. And so we can help your team, or Devin can actually, he's actually a better person. So definitely reach out to Devin and we can definitely disseminate and share information on what those passing metrics would look like for our assay. With that being said, I'm going to pass it over to Aisling, who is our product marketing manager.
Thank you, Hoy. So yeah, hi, my name is Aisling, and I've met some of you throughout the day, but I actually got started in the lab. I was in an evolutionary development lab where I looked at the reversal of the anterior posterior axis during development of a marine flatworm. And then I just jumped into industry, and I've kind of been there since. So with that, I've worked with CRISPR, single cell and single cell spatial proteomics and transcriptomics technologies. And today, I'm going to walk through a little bit of automating data analysis with Trailmaker. So we've seen today, and we've definitely been focusing on fixation and barcoding and library prep and how we're automating those steps, but I'm going to focus on Trailmaker and data analysis. So for Trailmaker, we've really designed it with the intent that it is very user-friendly and it will make single cell analysis widely acceptable. It has flexible data import and export options, and it enables efficient collaboration, and it's free to all academics and Parse customers. So taking a look at what Trailmaker does, it's split up into two different modules. So the pipeline module takes those raw FASTQ files and processes them. And then the insights module goes through data exploration and visualization. So fully taking you from those raw FASTQ files all the way to publishable figures. And let's take a deeper look at the pipeline module. So you can see the video behind me, but imagine you've completed your Evercode run. You automated it. It was super easy. You got more hands-off time and you have your sequencing data. So you're going to take those raw FASTQ files, begin by creating a new run. It'll prompt you with a couple of questions just to get some background information. For example, what product you were using, what was the chemistry. You will upload your sample loading table and select your reference genome. The reference genome will be in a drop-down menu. And if you don't see your genome there, it's no problem. Please reach out to us at support, and we're happy to help you upload the genome you need. So you will click run, and then it will generate this summary web report. From there, it'll give you a couple of different metrics, as well as the heat maps of your plates, which will enable you to just do some QC of your run super, super easily. And if you have if you want to walk through this with anyone, you can always reach out to your field team and they'll be happy to walk through the web report with you and answer any questions or do any troubleshooting. And from there, it's really easy just to click on over to the Insights tab. You don't need to do anything else. But if you do already have your process data, your count matrix, you can always upload them into Trailmaker and then go to the Insights module. So jumping into Insights. So there are 3 different components of this, starting with data processing. So here it's going to walk you through 7 different steps to process your data. It's very transparent. It'll tell you what it's doing at each stage. It will also automatically apply filters by default. You can manually overwrite that at any time, depending on the biology of your sample. From there, you can easily cluster your data based off of whatever clustering settings make most biological sense to your sample. And you can hop into the data exploration tab. So here it will enable you to automatically cluster, annotate your clusters. And then just to verify that, you can always take a look at the heat map below, look at the gene expression of a couple of your marker genes, and just validate that the annotations are as you would expect. So I'm going to kind of give you one example of something that you can do in Trailmaker, but there's so much more. So we are always happy to kind of walk you through a more fuller demo at any point in time. So say, for example, you've selected your cell types of interest. You want to go and look deeper at monocytes. You can take it over to the Genes tab, which you can see on the left, and do differential gene expression. So for example, look at monocytes versus the gene expression of the rest of the data sets and see which genes are most significantly either up or down regulated. From there, you can select those genes and take them into plots, select a dot plot and run through that and copy in those selected genes just directly into the dot plot. So really focusing in on what you're interested in. And the dot plot is fully customizable. You can change the colors, the scale of it. Also, you're able to directly export it and put it in whatever presentation or publication you're working on. You're also able to export that data and put it in another tool if you'd like to do any other analysis. And with Trailmaker, it's quite easy to collaborate. It's literally just clicking the share button and adding someone in on. So you can do this either on the Insights experimental run, or pipeline, or both. So really facilitating the collaboration between wet lab researcher and bioinformatician, or core and the researchers that they support. Or maybe your collaborator is across the US and making it easy to collaborate that way. So hopefully I've gotten you a little bit of excited in Trailmaker, and you want to get started. So we have a couple of different ways you can begin exploring. So I have the QR code up on the screen, which you can scan. It'll take you to the Trailmaker webpage on our site. But it's also really easy to find underneath resources on our site as well. From there, you can go to any one of these links. So you can create an account and just get started with analysis today. Either upload your own data that you already have, or we have Parse data sets that you can dive in and start exploring. We are, as I mentioned earlier, also happy to take you into a more in-depth demo as well. Either reach out to support or connect with your local field team, and they'll really be able to walk you through everything. And I know a couple of you already were able to see it upstairs today. And then finally, if you're not quite ready to jump in Trailmaker, but you just want to learn a little bit more, we do have a course on mastering single cell analysis, which will be a deep dive into each one of the parts. So you can learn more that way. And then finally, so hopefully you're ready to jump in and join the ranks of Trailmakers out there. But there's and able to see how easy the tool is to navigate, how quickly you can do data analysis, and then generating those publication ready figures. Okay, and with that, I'm going to invite the other speakers back up, and we can open it up for any Q&A.
I mentioned the temperature for the cell is very important, right? So do you think for the passive or whatever you call the cooling module, so how do you control the temperature? You're going to do some temperature measurement, or I know certain devices they have active feedback function.
Yeah, I think like for, I think it's different cases depending on what device you select. Like if the passive cooling where we basically filled like a block with ice and shove it in the freezer. We actually had a probe to measure how long it takes to roughly get to the temperature that we want. And then from there, we kind of monitor how long it's good for before it goes past our temperature threshold. With other devices like like an automated device, you can definitely set those temperature and
Yeah, as like for
yes, exactly. And that's a nice thing about like an automation system versus the passive one.
okay, thanks.
Yep.
So I don't know who this question was for, probably Devin. So I've worked with liquid handlers in the past, and I think this is great, obviously, because you want it to be predictable, you want it to be consistent, human error is a thing, but robots crash.
Absolutely, yeah.
And so what happens when you have a mega kit on a robot and it like
Yeah.
screws up?
No, for sure. So one of the things internally that we've done when we've automated the Parse workflow is make it modular in nature. So there are about 21 scripts that encompass the entire Evercode process that enable that type of situation from, being preventative of you continuing on should something happen. Another thing that we do, Hoy and I, in past lives, we build in error handling on the script itself to help mitigate the chance that something happens without you knowing, whether it's CLLD or it's a plate, for instance. I'll take the core paddles, pick up your deep well, put it down, boom, check to make sure it's there. Because a lot of times when you load the instrument, the error-prone situations are when a user misloads.
Yeah.
But the script itself, should everything be loaded correctly, you shouldn't encounter any crashes. A lot of that happens in the development, and our script, for instance, Hoy has done an amazing job. Like the little with the magnet, he should have showed that. But when I saw that, I was like, "How did you do that?" Like he said, it's all in the code. He used
Yes.
advanced scripting. So what I would say is our particular workflow, we've got it modular. If something does happen, we can help you recover from that. But upfront, we've got loading prompts that help make sure it's visual and walks you through that upfront loading. And that's the best we can really do.
Yeah, I think I can elaborate on that too. There's more options depending on how automated and how foolproof you can go into like a rabbit hole with this. And this is what I've done in the past too. One thing that I haven't, I want to dabble in is actually vision-based software, where if you attach your camera to an automation deck, it can basically look at pixels and determine if a plate is there or not. Or where the tip is, or there's certain things that you can identify visually through like software. And that's something that you can dabble in. We haven't gone that far yet, but definitely we're here to help if that's what's needed.
Yeah, I'm sure that a lot of the instruments and even the software have gotten better from the days when it would just gap and
Yeah.
crash tips and stop running, but
Yeah, but for the most part, we try to program things very in a module format so that recovery is easy.
thanks.
Thank you. So I have a question to Trailmaker. So we know that for like using the RAS to do like the UMAP clusterings, those often takes a lot of computation resources, and especially when increasing the cell number to like more than 10,000 or 20, 30, you need more and more computer resources, more time. So currently, I guess Trailmaker is running this on cloud, and then how long do you usually to regenerate a new UMAP? And is a maximum number of cell numbers you limits for the UMAP, something like that?
Yeah, so we're working on increasing the total number of cells that you can run with Trailmaker. But you can run through the Evercode T output, no problem. And then for the second half of how long it takes, it's really quite fast. The only thing with that is it's going to keep your experiment in pipeline. And then so you've I think it's 30 days to either download the full data, but it'll be in Insights. Does that answer your question, or is there a follow-up?
I can add a bit more too as well. One thing also that I think Aisling mentioned, but is quite helpful to mention again is that you can actually import and export Seurat objects directly into Trailmaker too. So it actually makes it super easy if you've been dabbling in bioinformatics or you have a lot of experience in bioinformatics and you rather do a lot of your work in Seurat, but then want to put it in a Trailmaker to make it easy to analyze for, say, a collaborator. Super easy to actually import and export Seurat objects during the analysis, which is really nice. And the other thing is that it is on the cloud, and the cloud analysis team that's built this has definitely optimized a lot of kind of the back end to try to make it basically as fast as possible, which can be different than if you're using like a local computer when you're running Seurat kind of separately. So that's definitely one of the benefits of basically working on the cloud with kind of our kind of that back end.
So for the workflows on the various instruments, is Parse going to be representing as far as the development, implementation, and scaling of the process for the various instruments, or is that done by the manufacturer of the instruments?
It's a good question. So the approach that we're currently taking with the vendors is essentially collaborating with them and consulting them on the workflow so that they can best optimize it for their instrument. They're the experts on their instrument. They know the limitations, the capabilities. We know the science. And so that's kind of how we're approaching this. We're letting them make the best effort towards automating our process, but we give them the tips and tricks that we've learned when we've developed in-house. And so essentially what you're going to end up with is for OpenTrons, for example, or Integra, they've automated the method their way on their instrument, and they would sell you the components in the instrument as is to run our method. Should you say, hey, OpenTrons, I want to run Parse, they'll be like, hey, he's actually got the printout right there. The cost, the modules that you need to run it, and you'll take it from there. Now, support-wise, any hardware or anything like that goes wrong, a crash or something, you're going to contact OpenTrons or Integra. But if it's scientific, if it's chemical, biological, mishaps in the data and the analysis, you'll contact Parse, and we'll deploy and try to see what might have happened in that workflow, what you can do to improve, the upfront fixation part, because whatever you put in kind of affects what you're going to get out. So we'll consult you there, but mostly what'll happen is you'll interact with the vendor themselves on any of the hardware and mishaps that you might encounter. But again, we're collaborating up front to make this pretty streamlined and foolproof. So should be good.
Of videos and talks. Thank you, Lord. I wanted to ask about that, I actually, too. But the first one is, are those tips and tricks, like those cool things with the magnets, or with the fixation dispensing, are those built primarily into the protocols that you've been developing on the Hamilton and the Integra? I.e., are those also built into the Optitrons protocols?
Yeah, I think we've shared it. Basically we've shared all the tips and tricks with our partners. And at this point, it's more translational, right? They definitely have like liquid class, like speeds and volumes, those type of things that we can translate to them. And they can basically, there are different settings, of course, for the instrument on how to achieve those. And that's what we're after.
Yeah. And then I guess the second thing, which is kind of a comment, I think the Trailmaker cloud thing is very interesting. It would be really great to see, maybe this is aspirational, a kind of cell by gene version that's all parse based. That maybe that we could start referencing.
Sorry, I missed the, a what?
Like a database repository all
Oh,
parse runs and be
yeah.
like some annotations. So that we can stack these data sets up and create like parse
Repository.
generating repository of data.
Yeah, that's a really neat idea.
Yeah, well, and like one thing I think it's worth pointing out on that front here is we already do have parse data sets that are up there. So like Ashley mentioned in the presentation, in the insights module, if you don't have like your own data set to ready to analyze, you can actually go in and just click on preloaded data sets on like PBMCs or heart or whatever tissue type you're interested in. We're basically continuously uploading more data sets up onto that insights module. And then kind of in addition to that, we're also working on basically additional ways to have integration of single cell data sets on there. So, for example, you can upload like data from a different 10x, from a different single cell technology like 10x or BD Rhapsody or something. If you want to kind of integrate all your data into the platform, you can take more parse data sets, you can take data sets from other technologies and actually integrate them all in Trailmaker to basically do your final analysis. Because ultimately, if you've got lots of different data sets, but you want to analyze everything all together, you want to have all the data in one place. So we want to basically facilitate that as much as possible.
Hi, I'm Mark from Shape Therapeutics. I'm a huge fan of the Evercode kit. I've used the mini and the wild type. I've been very pleased with the results. My question today is with the Trailmaker. So when our AI team had generated, so we had our own process for generating the cell by gene matrices. And because we did a bigger NovaSeq run, they did encounter like, we also wanted to put it on the Trailmaker for me to play around with the data. And we ran into an issue where there was, I think, a limit of 15 GB for the CRR object to be uploaded. But our file size was like 90 GB, so we had to downsample the CRR object. So is there some solution for that?
I can speak to it. Yeah, so it for some of the data sets that are much larger, I can tell you we're currently developing kind of a next version to basically support significantly larger data sets. It does support up to 1 million cells, but it can depend on if you have like on how much how dense the matrix is. And so if you and if you go over 1 million cells, I think that's where we're basically currently developing basically a kind of next version here that's going to allow you to go faster or faster and support larger data sets to essentially allow you to say integrate data like I was mentioning earlier. So it's definitely in the roadmap to be kind of have a next evolution on that. Yeah.
Johnny, to your point, back on the tips and tricks. Once you get the instrument, if you're seeing, personally in your lab that what was built isn't really achieving what you need, we're here to consult you. Like, that's my job. So I would make sure that we collaboratively address whatever the situation is, and you'll have a personalized solution to your cells that you're running. But again, what you'll have is a baseline ability to run samples, and then when the data comes out, then we'll address everything from there to get you where you want to be. So again, my main role, so if you are running into any problems with automation, contact me, Devin at parsebiosciences.com. And we will go from there. We'll sort it out and we'll get you up to where you want to be, because again, everyone's got their own cell type. You're gonna get different success metrics for your own personal laboratory. So we're developing what's optimal for us. It might not be the same for you, but the automation is there and it's solid. So then we can make small tweaks to help you get where you need to be. Some people put the Integra in a deli fridge to keep the whole environment cold, because cells like the cold, so there's things like that can happen for you specifically that other people might not have to do. But again, we're here to help you get up and running and get the data that you want. So